Welcome to Tracing Your Family Roots. I'm Arline Sachs, and today my guest is Dick DiBuono. Welcome, Dick. I'm going to talk about using DNA for finding family, and I have Dick here because he's just recently had his DNA tested, so I'm hoping he can help me in explaining some of this.
Certainly will try.
What I want to talk about today is, first of all, the generalities that that you find in DNA. Then we'll talk about the National Geographic groupings called a haplogroup, a little bit about the biology, the specifics about DNA, some of the samples, which I see is spelled wrong there, sample results for the two groups. I don't know how that C got in there. And then we're going to look at three different results. The Worth group, this group by Sidney and Richard Dick, your test results.
Okay.
And if we have time, we'll look at some police testing and paternity and show just how very, very different that is. And we're really only concerned with the genealogy part of it today. Now, human beings have 23 pairs of chromosomes, and this is just sort of a picture of them. And what we're really interested in is only this little tiny Y male chromosome. You know, a man gets the Y chromosome exactly from his father, and he gets an X chromosome from his mother. So you could really have both tested. Did you have both tested, or just the Y?
Just the Y.
Just the Y.
Just the Y.
At this point, we really can't tell too much from the mitochondrial testing. I imagine at some point, I still am hoping that at some point
it'll become important, right?
I'm hoping it'll be as important as the Y chromosome it has been for genealogy.
Well, the Y is the one that the geneticists take greatest stock in for genealogical purposes, because unlike the mitochondrial, which does not mutate very frequently through history, the Y has, and that's how they can separate out various ancestral lineages, so-called haplogroups. Yeah.
And you need to say that when there is a change, they consider it a mutation.
A mutation.
So that theoretically, you and a brother would have identical mutations.
Right.
Identical haplogroups should be exactly the same. If you don't, then there's a mutation.
Right.
And this is what they trace. They figure a mutation happens only every few generations, and therefore they can trace back. If you have somebody with the same designation,
sure,
then you've got a
relationship.
Yep. Now this is what was done, one of the first things that were published, and this is the DNA that was done by the National Geographic, and Family Tree DNA is the one that did all that testing, and we'll be talking about them as the testing. But this shows, what this essentially shows is that we're on, and I don't know if you can see all of the lines, but they all originated here in this general part in Africa. And you can see some of them, like this E group and the A and the B group. Well, the A and B is only in Africa. Mm-hmm. So, yes, you certainly find American of African descent that would have it. But
right,
as far as native peoples, that's only found in Africa. And it seems as they moved out, whether they changed to adapt to local climates or something like that. And they haven't, I just recently read an article where they're speculating on that, but I don't think they have any conclusions. Now, I'm going to talk a little bit more later on about this J, which is found around the Mediterranean, which is generally Semitic, and you belong, I think, to this I.
The I group.
The I group, which is up here in Europe. Now, what they do this... there are a few hundred major haplogroups and subgroups, and they're based on their Y chromosome SNP. Mm-hmm. And this SNP is the slide that's up here now, and it's those spots that change very, very slowly. It's a single nucleotide polymorphism, a very slowly mutating location that's used to define the haplogroups. It's a whole big explanation, but it just means that this tiny, tiny spot is
that's
changing.
The indicator.
That's the indicator that we're looking for. Mm-hmm. So that's how they get all of these different color lines that you see on the screen. And you can see this one, which I find is very interesting, this C group that went around what was probably a landmass going through the tip of lower India, down to Australia, and then all the way up along the Pacific coast, and then down into the Americas. It's
Fascinating.
yeah, I think it is. And then the Q group, which came from higher up, that's the one that ended down in South America. I noticed they don't have anything over here in Eastern America, so I don't know whether they haven't tested there or what. This, on the other hand, is what they figure the mitochondrial, or the women migration was. And you'll see that there are many more that seem to have survived coming to the...
Right, you might want to point out too, Arline, that even though they use similar letters, the letters are not compatible between the mitochondrial and the Y DNA as far as identifying haplogroups.
Yeah, that means nothing. It's just it's just like giving
nomenclature.
It a name. Yeah, and the Eve is where they figure that everybody is descended from someone here. Some of these go back more than 100,000 years. Again, this L, which seems to have stayed in Africa, seems to be only down there, and then some of the others came over here, and so on. Now, we're going to talk a little bit about the DNA itself, and it keeps getting smaller and smaller, and to me this is very, very hard to visualize. We've seen the chromosomes, and the chromosomes are found on the genes, or on the X, the X and Y chromosome. The average chromosome is 5 centimeters. Now, if it's 5 centimeters, how can we can't see it? I mean, I can't.
Well, it's very small. Yeah.
And the chromosomes are made up of the genes, and there are different chromosomes. We saw that, there are 23 pairs. Chromosome 1 has almost 3,000 genes, and the Y chromosome has the fewest at 231. And the DNA is then found on the genes.
That's
so you
correct.
keep getting smaller and smaller and smaller. And since we can't even see a single cell with a naked eye, it's just really hard for me to picture this. Total length of one adult human DNA is 2 to the 10 to the 13th power meters. That's equal to the sun.
And back.
It's incredible. It's just, now, they're bases, and the average gene has 3,000 bases. Largest gene has over 2 million bases, and 3,147 million nucleotide bases.
3 billion, over 3 billion, yeah.
These little tiny things. Now, you want to explain what you had to do when you were tested?
Oh, I'd love to. I use the Family Tree DNA commercial testing organization, which seems to be the most prevalent one in the genetic field these days for genealogical purposes. And they sent me a kit. The kit contained 3 plastic scrapers that are not sharp, but they're very mild. Yeah. It can be seen on the screen here.
It's just like a tiny toothbrush, really.
Right. And I took the first one to the inside of my cheek and vigorously scrubbed it for about a minimum of 60 seconds is what they asked to do. At the end of that point, they have 3 vials that accompany these 3 scrapers, and you discharge the scraper end from this device into the vials, which is a preservative, I presume. You cap it. Yeah. You do this 3 times. You have to wait at least 3 hours between scrapes, and then you send it off and wait 4 to 6 weeks for results.
Yeah.
And
you get very impatient waiting.
Right.
Okay, before we get any further, I want to say that what we're using and talking about today is for genealogy purposes, and there are lots of other purposes to test for your genes and test the DNA. And so for medicine, for police work. We're not really talking about that today. We're talking only about the DNA. The genealogy
for testing for genealogy.
And I'll show how the other one goes, a very brief example, only if we have time later on. Now, you mentioned that you use Family Tree DNA, and they are, in fact, the ones that did the National Geographic testing.
Right.
This chart house.
They're the ones doing all the testing for the Genographic project. National Genealogical.
Some companies are just going to give you like letters A, C, G, T, whatever,
right.
and then you're sort of stuck with what does all of that
right.
mean. Family Tree does have a database that you can look at, and even if you haven't been tested by them, and they've set up like a bulletin board type thing that you can
right.
ask questions on.
And check to see if you have any matches out there in the world.
Okay. Bennett Greenspan is the CEO of Family Tree DNA, and we have done a show with him about 6 or 8 months ago. He did it. He got into this because he found somebody with the same last name living in South America, and he was trying to figure out whether they were related, and he'd heard about the DNA and heard that you could do this, but he couldn't find anybody that'll do it for him. And then he contacted Mark Hammer of the University of Arizona, and Mark said he'd do it for him. And Mark also said, you ought to start a company that does this. And so the two of them now, he's the head
scientist
right, with
for
Family Tree
DNA.
And so that's how this company got formed. They were really formed in order to do a service for the genealogy community.
Right.
And all of the other companies said, oh, this is a good idea, and then try to get into it. But if you use a smaller company, the disadvantage is you don't have this large database to check on.
Right.
Family Tree has over, and it's been a couple of months since I checked the numbers, over 120,000 people in their database, and he doesn't list it on the web of how many Jews he's tested, but it was over 4,000 as of last fall. So that's just a number I have. The more people that you have tested, the greater is your chance of matching up with someone.
Right.
And
I keep looking.
You keep looking.
I haven't matched yet, but we'll talk about that.
Okay, I want to talk about a few specific examples. And this was a little bit over 2 years ago. A local newspaper, the Washington Jewish Week, had an article about DNA. And the reason what caught my eye on it was it mentioned 3 people, and one of them was Saul Issaroff of England, and I happened to know him. I said, why is his name here in this newspaper? And as I read further, it was about a Puerto Rican woman named Sonia Rosa who found that her father belonged to the J1, J2 haplotype, which is the Semitic one, which I pointed out came from around the Mediterranean, more or less. And that he was related to these Saul Issaroff and another one, Herb Upshur, and they really didn't know. They were sort of surprised at this result.
Sure.
It's really surprising because her father was black and had absolutely not the slightest idea that there could be anybody Jewish in his family. And his mother was, she also had her mother tested for the mitochondrial, and found that her mother had American Indian ancestry, although her mother was white. So she said, "This is
a fascinating set of results."
Yes. So as we got to trying to figure out how this could be, it must have been that one of her ancestors had been a converso or a Marrano or a New Christian. I don't know if you're familiar with any of those of those terms. In 1492, all of the Jews were expelled from Spain. They went west to Portugal. They went up to Holland, what's now to the Netherlands. They came to the New World, mostly in South America to begin with, and the islands. So there they are in Puerto Rico. Would have been one of the islands, logically, that these people would have come to.
Sure.
Her family came from the far northwestern corner, meaning they were trying to get as far away from the Spaniards as they could. And she said she just grew up there. You know, they were farmers, and so it sort of all makes sense. It's a logical. I mean, we have nothing to match it. Also, she became part of the a group called the Worth Group, and the Worth Group is simply 5 initials of the first 5 people that seem to match. It matched to some extent, and
So they joined
The-
in
yeah,
a
they
group
began
to
to
follow
study.
their genealogical relationships.
to try to feel,
Yeah.
and you'll see as we get through this is what we have to do.
Sure.
Now, it was started by a man by the name of Herb Upshur. Now we'll get to this slide that I have sitting up here. And it's called Table 1, and it's Herb Upshur, whoops, sorry. And a man named Hipshire, and he was under the assumption that, gee, those names are pronounced
Close.
very much the same, because his name would have been pronounced like that.
Originally?
Yeah.
Yeah.
And so he asked this man to be tested. And if you can see these numbers on the first 12, you'll see they don't match at all. And before I go further, I think I need to say that when you get tested for DNA, you can pay for 12 or 25 or 37.
Or all the way out to 67,
67.
as I did, Arline.
Yeah.
Of
Well,
course, the costs go up proportional to the number of markers that you ask for them to test for.
And that's
Yeah.
why they generally suggest if you think you're related to someone, just do the first 12.
First 12.
And if you have 11 or 12 on that, then go ahead and do further testing.
Sure.
On the other hand, if you're a genealogist and, like you think that eventually you're going to have to get up to the 67, you might as well go ahead and spend that money.
That's why I did that, and also for whatever purposes it can do to benefit the entire genographic program.
Yeah.
Offered those up.
So anyway, he did this with Mr. Hipshire, and you can see for the first location they tested was 12 and 13, and for the
Yes.
next one, 23, 22. Obviously, nothing matched.
no.
Oh, well, here there's a match, but
Right.
that means nothing.
Doesn't mean anything.
So they were not related. But he found out that somebody else by the name of Saul Issaroff in England matched him. And here they had different
Jeez.
last names. And you can see on the 37 one that they did match perfectly. Not only that, they do have some anomalies in this
23rd marker?
Yeah, they have 13.1, which
It's
is
a mutation.
It's a mutation, and it's an odd mutation that they obviously both have.
And share.
And also they seem to have two 26s, which I don't understand either, but there's a 16 here
And a
and another
10.
26 here that says 10. And
Those are the number of alleles, or the
That's-
repetitions of the bases
Yeah.
at those marker points in the DNA, as I understand it. I'm no
I think
expert, Arline.
yeah, that's, and I don't understand how they repeat
That's
and all
just
of that,
from
but
the
they
reading.
said that's the
Right.
repeats that they
Right.
have. So obviously they matched, and then he went on further. And this is genetic distance of the various families. Now he's up to over 60 matches. Wow. And
I envy him.
and this is what they found. He has two people that match entirely at the 67. So these people are obviously fairly closely related,
Right.
probably no more further than third cousins, I would guess. They have six that are only one position off. They have 10 that are two positions off. And I want you specifically to look at this. I think 3 is what Saul Issaroff ended up being off of, although they matched perfectly on 67. Right. When they got to, on 37. When they got to 67, they were actually 3 off.
3 off. Huh.
And we'll see what happens when you're looking at something like that. You can develop here what they call a phylogenetic chart. And this really confused me. Why are this Berger and Tannenbaum, what they call modal, I'm not sure if you can read those words on the TV screen, but they are the center. And what happens is they are the one that have the most common to everybody else. Just like in Africa, they had the Eve, or
Sure.
they think that it came from there. This is what has the most common. Issaroff is way up here, and here are the other two that matched completely. Now, in order to help me understand it, instead of doing it on 12 or 37 or 67, I tried to show it here on the basis of 5 people. Let's say person 1 tested A2345, and person 2 tested 123B5. Well, if you compare these two, out of the 5, they're different by 2 points.
2 markers.
And person 3 was I had him at another place. And here's the modal, 1, 2, 3, 4, 5. If you compare person 4 to any one of the others, they're only 1 off.
Interesting.
So that person 4 would be the modal, and the other 4 would be lines out from him.
Emanating. Okay.
So that's how
Interesting.
they get this, but it's obviously more complicated because they're
yes.
testing it on much more. Here is Sonia Rosa. She's way off here. But, you know, still similar enough that she has to have been descended from whoever Berger and Tannenbaum
We're descended
were
from.
descended.
Yeah.
And then my next question was, wait a second. If these are all directly off of it, you said there were 5. 1, 2, 3, 4, 5, 6, 7, 8. There's 8 people here. And then he explained further that I was in touch with Herb Upshur many times, and these charts do come from him, so I do want to give him credit.
Sure.
He explained that the differences between himself and Issaroff are not common to anybody else, and that's why the differences are the difference from him from the modal, and his mutations don't match anybody else's mutations. And that's why he's on a separate line.
Separate line, I see.
It's a long line to indicate that there were 3.
Yes, there could be others, yeah.
So, you know, maybe they should have a dot here and dot here to indicate that there were 3 separations, and that's what happened also on a couple of the others. These 2 matched each other, but again didn't match the others, and that's how they get to have all those separate lines. But and this is what you can tell if you match 37 out of 37, which is, remember what Issaroff did.
Yes.
There's a probability, 50% probability that they lived no longer than this number of generations.
The most recent common ancestor.
Yeah, the most recent common ancestor.
Right.
Or 95% probability that they lived no longer than 7
generations ago.
Now, because you have all of those different last names, we can sort of guess that it was probably at least 200 years, because that's the time for Jews that they took different names, or that they took last names at all.
At all, right.
In Eastern Europe, they did not. In Germany, they might have had names earlier, but or in France. But the further east you got,
they usually didn't have surnames.
They didn't have
until
surnames.
About 200 years ago.
Until Napoleon decreed it, and then as his reign spread eastward. Now there are a few exceptions. In Austria, they started in 1790s. So there are a few pockets where a duke or something insisted that they have last names for taxing, of course.
Right, right.
It helped them take.
Always economic motivation.
Yeah. So if you're matching only 35 out of 37, you know, it might be as many as 14 generations. So that's 400 years ago that you're talking about. And this is on the basis of only 37. I don't have this kind of a chart for 67. I haven't seen it yet.
Yeah.
Somebody else could.
I haven't either. I have seen that on DNA's on Family Tree DNA website.
I think that's probably,
Right.
But I don't know why they haven't done one for 67 yet. Okay, now the second group I wanted to talk about was Sid's, because his situation was a little bit different. His father was one of 10 children, 9 of whom survived to adulthood, 7 males. But of those males, Sid is now the only male, and of those, he's the only one that had male children, and we only have 1 grandson. So all of those go down to 1. And he has been looking for a long time for anybody with Sachs. Well, when he had this done, lo and behold, he did match other people, but not one of them is a Sachs. So again, there's theories as to why this might be, but I don't want to spend too much time with it. But he has, formed a new group of, and one of the things that Bennett Greenspan happened to look at this when he was here, and I brought the certificate as an example. He says, "Oh, this location 455 has a number 8 in there. That's very unusual." So that's he's, oh, well, if that's unusual, let's start a group. And, you know, so he
Sure.
has found a group, and they have some things that are similar. And again, they're Kohanim, which among the Jews is something sort of special. It's something that people would know because it's always passed down from father to son as a tradition.
Mm-hmm.
And so most men, if they're still active at all, would know it. And I said, really didn't. His father didn't, but we found out for sure that he is through other things. So this gives us something to look at. Now, what you have to do after this is get all of these people with all of these different last names and find out where they're from. And they all are within a couple of 100 miles of Kovno, Gubernia, which is like saying they're from the state of Virginia or the state of Montana. It's a large area, but it's an area. It's an area.
Yeah, definable area.
But nobody knows where.
Yeah, okay.
And some of them just have a general vague idea that maybe it's Lithuania or maybe it's something, you know, one of the countries nearby there. But this gives us, what it gives us to work on is that's what he has to, you know, they have to work together.
The group has to work
now,
together.
yours is a little bit different. We're almost out of time, so do you want to say what?
Well, yeah, I was surprised by my Y-DNA test results because I turned out to have the haplotype that fell into the I2b haplogroup, which is not one that's very prevalent in Italy, although both my paternal and maternal ancestries are from Italy, and I've traced my ancestries back, my paternal line, to the 1600s. But this group, according to Family Tree DNA, authorities indicated that it started somewhere around 22,000 years ago in northern France, and today populates mostly Scandinavia, Saxony, and Germany.
So it's got to be
very few in Italy.
So it's got to be some conquerors or Vikings or something.
I suspect the Normans.
But we're almost out of time.
Okay.
But that's interesting, and so now you know that you have to start a group or look for those that are from there.
Right.
I just want to leave this last slide on. If anybody wants to look at some of the other shows that we've done on DNA, we have had Bennett Greenspan on, who I've mentioned is the CEO, and we've had Herb Upshur, and we've had Sonia Rosa. So check those out, and check out FamilyTreeDNA.com. And thank you so much for
You're welcome, Arline. This has been fascinating. I've learned quite a bit.